Hello, my name is Sydney Gordon, and today I'm going to be talking to you about the enzyme telomerase reverse transcriptase. This enzyme is a catalytic protein, as all enzymes are. It starts off as a DNA sequence, and it's transcribed and translated into a functional product. Once the product is made, what it does is it, in turn, catalyzes the formation of telomeres. Telomeres are a sequence or a dummy sequence of DNA nucleotides that are added to the end of chromosomes, so that when the cell undergoes division and subsequently replication, there are no important facets of genetic information that are lost, because every time a cell replicates, there's 50 to 200 base pairs that are lost in that process. So when you extend the length of a telomere, it ensures that if that telomere portion is not translated or is not replicated, then it's basically no harm, no foul to the body, because the important information was translated in that process or was maintained in that process. But that's how it works in the body. In the brain, it functions very differently. In the brain, telomerase reverse transcriptase has been proven to curb some of the effects of neurodegenerative diseases such as Alzheimer's and Parkinson's. To give context to this before explaining how it does that, there are proteins in the brain called alpha-synuclein, and they're harmful because they form Lewy bodies in high concentration. These Lewy bodies have been linked to Parkinson's and Alzheimer's pathogenesis. Under normal body conditions, the cells in the brain can metabolize the breakdown of alpha-synuclein through three main processes. The first is through the formation of a ubiquitin proteasome system. The second is through CMA, which is chaperone-mediated autophagy. And the third one is through macroautophagy. What TERT, or TERT, or telomerase reverse transcriptase, targets is the latter of the two. In explaining what the two processes are, CMA is related to, or breaks down the monomers of alpha-synuclein under normal body function, and macroautophagy breaks down the polymers of alpha-synuclein. So what it does is it takes the cell, the brain cell, merges a vesicle into the body of the cell, the cytoplasm, takes the alpha-synuclein inside of a vesicle down microtubules into the cell, where it's hydrolyzed in a lysosome and therefore rendered unharmful to the body, to the brain. Be it mutations or through old age, the effects of alpha-synuclein increase because the systems that catalyze its breakdown begin to fail. What research has discovered, though, is that with the increased presence of TERT in the brain, there's an increased ability of those previously failing autophagy systems to help break down that alpha-synuclein. And in turn, what that causes is an increased effect on motor function and an increased ability to remember things, recall, in Parkinson's and Alzheimer's patients.